Once again, welcome beta. In that continuation of the animal cell culture, today we have chosen the topic what are the equipments are required for the culturing of animal cell, and moreover you must know that to culturing the mammalian cells or any other living cells which is taken out from the animal's body required nutrient medium. It required the constant temperature, aseptic condition, that is the sterile medium without contamination or without the infection. And one of the more important things that actually if you are taking the animal cells from the warm blooded animals, so that constant temperature should be given to the cells which we are taking for culturing purposes. So there is a one of important thing that we are taking 5% of carbon dioxide along with the temperature 37 degrees centigrade. So this mixture of the gas plus temperature is more suitable for the culturing of the cell because our body temperature is equal to this range. So another important thing is that what are the main important equipments are used in culturing of the cells are as follows. So first one is the culture flask. This is the culture flask. Already you have seen or you have been aware about the culture flask because in the tissue culture technique these things we have already discussed. Apart from that, petri dish, you are also aware about the petri dish. Now the multi-well plates. Actually multi-well plates are made up of the glass or the superior quality of the plastics, and there are a number of columns. Instead of one petri dish, there are some of the blocks like this. Therefore, we call it the multi-well plates. And these plates of the petri dish are used to keep the nutrient medium, and over that the culturing cells will be placed. Now, cell scraper is because of it is used for the purpose of the picking up or putting the cells over the nutrient medium or the culture medium. Now the basic important equipments come from the tissue culture hood. Tissue culture hood is what? This tissue culture hood, it is also known as the laminar airflow hood. What is the meaning of laminar? When there is a continuous movement of the air or the fluid in a multiple layer, it is not through the direct air moving and reaching to the points. It is not so. When the air or when the fluid is moving through a layer, suppose this is one layer, then second layer, then third layer, then fourth layer. So in this way, this type of movement is called the laminar air flow hood. Actually, tissue culture hood is also known as the laminar air flow hood. And what is this tissue culture hood? Actually, tissue culture hood is a well-equipped enclosed cabinet, or you can say that it is a equipment that is designed to prevent the contamination of the biological samples. Biological samples means these are the cells which are taken for the purpose of culturing. So this is the biological sample, and it is also have some devices that is kept inside the hood, and the main role of that device is to wipe out the air outside from the culture vessels or from the tissue culture hood. And there is no the air which is filtered in the tissue culture hood is not a normal one. It is not a normal like a particle filter or the air filter, and that one is its name is the HEPA. What is meaning of HEPA? It stands for the high efficiency particle air. So HEPA is also installed or it is also fitted in the tissue culture hood, and the main function is it continuous outflow the foul air which is inside the cabinet. So this continues throughout the air outside with the help of the HEPA, that is high efficiency particles filter, which is also suited fixed or that is installed inside the cabinet hood. Now the another function of tissue culture hood is what? It has two basic roles. It has two basic functions. Number one function is that it is made for the purpose to make the environment sterile. That is, protect the tissue culture from the operators. Operator is here. The person who is working on the tissue culture or the growing the culture is known the operators. So it make the medium sterile. Number second is it protect operators from the tissue culture. So the second one is that it is the main purpose is to minimize the chances of infection risk. So basic two function is delivered by the tissue culture hood. Number one is sterile environment, that is the sterile environment. The constant sterile environment is established in the culture hood, and the second number is it make the less chance of infection by the person who is working on tissue culture or culturing of the animal. Apart from this basic idea or the basic description, one of the important aspect of the tissue culture hood is that it has ultraviolet rays also. These blue light color ultraviolet rays is also given to the culture hood frequently for 2 minutes or 1 minute, and you know the UV rays is very essential for killing the germs or it stops the contamination in the culture tube or the culture vessels or tissue culture hood, and it is equally important to clean the inside medium of the culture hood. So this is about the tissue culture hood. So what is the tissue culture hood? Tissue culture hood is a equipment or it is a sealed cabinet, and this sealed cabinet is basically used for keeping the biological samples, and it is also having the HEPA. What is the meaning of HEPA here? It is a high efficiency particles air that continuously remove the air which is inside the cabinet. So the main purpose of this HEPA is filtering the air. Now another approach of this tissue culture hood is that it protect the operator from the tissue culture and it protect the tissue culture from the operator. So there is two things. One is that it make the medium sterile, and another one is that there is a less chances of infection risk from the operator to the tissue culture or the cells which are cultured. Now second one is the CO2 incubator. What is the meaning of incubator? Incubator is actually there is a CO2 incubator in the bio lab also it is there, it is biotechnology lab also available. So incubator is an apparatus or it is a sealed enclosed or the sealed apparatus where we keep the sample or the biological samples or the tissues so that the constant environmental medium remain for prolonged time period. And if you are asking about the CO2 incubator, then here what we are taking here, there are multiple chambers are there, and in that multiple chambers we are using the carbon dioxide here. So 5 to 10% of carbon dioxide is also added in that CO2 incubator, and the main purpose of the CO2 incubator is that it provides the controlled temperature and environment as it is found in the living cells. Already I told that if the cells are taken from the mammalian cells, so mammalian cells body temperature ranges from the 30 to 37 degrees centigrade. So being the warm-blooded animals, what we have to do, we must keep the constant body temperature. So the cells will have the same temperature and the environment. So therefore CO2 incubator is basically used for the creating the controlled environmental temperature as well as condition. Now in the CO2 incubator, the question is there, how we maintain the pH, how we maintain the temperature. So already I just told temperature is maintained by the placing the 5 to 10% of CO2 in that chamber. Apart from that, a pan of water is kept inside the chamber. That is CO2 incubator is having 5 to 10% of CO2 as well as a pan with water. The main function of this water is there. It creates the insulation, that is the artificial medium, and generate the humidity. If there is no humidity, if there is lack of humidity, what happens? There is the desiccation. So the main purpose of keeping a pan of water in the incubators or CO2 incubator is that it prevents the desiccation. Desiccation means dryness to the cultured cells. And how the pH is maintained? pH is maintained by the buffering, and that buffering is important, or it is given by bicarbonate or carbonic acid. And you should know that 7.4 pH is essential for the well cultured growth of animal cells. So once again here, CO2 incubator is an apparatus. It is a sealed apparatus which has a multiple chambers, and each chambers are designed with some concept or some principle. For example, if I are saying that there is a pH maintaining, temperature and gases maintaining, or humidity maintaining. So all these things are kept inside the incubator, and a pan of water is kept inside the incubator. Purpose is to make the real humidity that is outside. So outside and inside, temperature, humidity and the pH will be remain same. Now the pH buffering is done bicarbonate or bicarbonic acid, and the best temperature or the best pH for the culturing of cell is 7.4. That is slightly basic. It requires slightly basic. Now next one is the centrifuge. Centrifugation or centrifuge is a device, or it is an instrument or the equipment. Normally it is used in like this, in this circle way, but not in a very high speed. It requires a low speed at 20 degrees centigrade only. If suppose we have increased the temperature 40, or if you increase the temperature 30, and if we are doing the centrifugation at high speed, then what happens? The cells will be ruptured, their plasma membrane or maybe the organelles will be out. So while we are doing the centrifugation, so we must keep that in mind that 20 degrees centigrade temperature is required, and it will be in the low flow, that is the not in the high speed, that is the low speed. And the main purpose of centrifugation is that it separates the cells which are found in the cluster. So this is the rule of the centrifugation. Now next one is the inverted microscope. So what is the inverted microscope is here? These inverted microscopes are developed by the Lawrence Smith. Name is the Lawrence Smith. Yes, it is here. The name is Lawrence Smith. Lawrence Smith have developed this inverted microscope, and these inverted microscopes are really different from the classical microscope, that is a compound microscope or the simple microscope. What happened here? Actually, the cultured cells which we are taking on the petri dishes, they settled at the downside and nutrient medium is upside. Therefore, here in that inverted microscope, the source of light and the condenser is above. That is opposite from the classical microscope. This is because of that the cells are sink down. That is, cells are present at the bottom. Therefore, the source of light is inverted, so that we can visualize the picture well. So this is the inverted microscope. So this is about all the whatever the basic nutrient medium, condition, and equipments are required for the culturing of animal cells. Thanks to watching.